Hello everyone, myself Deepti Negi from MSc Microbiology second semester. Here's my presentation on the topic DNA replication. These are my contents. Introduction. DNA replication is the process by which DNA makes a copy of itself during cell division, or the basic definition of DNA replication is that it is the biological process of producing two identical replicas of DNA from one original DNA molecule. It is a complex process that takes place during cell division, that is the S phase, whereby DNA makes a copy before the cell divides through mitosis and meiosis. DNA replication occurs in S phase, and S phase is the synthesis phase of the cell cycle in which DNA is replicated. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. Now, let's discuss about the properties of DNA replication. The first property of DNA replication is that DNA replication is semi-conservative, which means that each DNA strand serves as a template for the synthesis of a new strand, producing two new DNA molecules, each with one new strand and one old strand. This is called semi-conservative replication. Next property of DNA replication is that replication begins at the sequence called origins. DNA synthesis is initiated by short fragments of RNA called primers. The elongation of DNA strand is always in the 5' to 3' direction. DNA replication can be unidirectional or bidirectional. If DNA replication is bidirectional, it means that it is a type of DNA replication where replication is moving along in both directions from the starting point. This creates two replication forks which moving in the opposite direction. And if DNA replication is unidirectional, it means that only one replication fork is formed. Replication is continuous on the leading strand and discontinuous on the lagging strand. And the last property of DNA replication is that new nucleotide strands are complementary and anti-parallel to their template strand. Next is the models for DNA replication. There are three models for DNA replication. These are semi-conservative DNA replication, conservative DNA replication, and dispersive DNA replication. As you can see in this figure, the DNA from the original double helix is blue in color and newly made DNA is pink in color. Now let's discuss about the semi-conservative DNA replication. Here, each DNA strand serves as a template for the synthesis of a new strand, producing two new DNA molecules, each with one new strand and one old strand. This is called semi-conservative DNA replication. Next is the conservative DNA replication. Here, it is an alternative method of replication, and according to this model, original double helix of parental DNA molecule is conserved. Two strands of a double helix of DNA remains joined to each other and individual chains remain intact. Hydrogen bond between DNA strand remains stable and original parental double helix act as a template for the formation of entirely new daughter DNA molecule in which all nucleotide subunits are entirely new. And the last is the dispersive DNA replication. In this scheme of replication, two strands of a parental DNA molecule totally disintegrate. Not only hydrogen bonds between two strands break and separate two strands, but phosphodiester linkage between nucleotide of each strand break and individual nucleotide disperse in the medium. That is why it is known as dispersive replication. Here the nucleotides are dispersing in the medium. From parental nucleotides and new precursor, two DNA molecules, two daughter DNA molecules are formed, and daughter molecule consists of the part of template parental strand and a part of newly synthesized DNA. So to determine which model of replication was accurate, a seminal experimental was performed in 1958 by two researchers. These researchers are Matthew Meselson and Franklin Stahel, and they proved that DNA is semi-conservative replication. Now the evidences for semi-conservative replication of DNA. Matthew Meselson and Franklin Stahel have performed an experiment using Escherichia coli to prove that DNA replication is semi-conservative. So what Meselson and Stahel experiment is? What they do, Meselson and Stahel experiment with E. coli grown first in heavy nitrogen 15, then lighter nitrogen 14. Here, the DNA grown in nitrogen 15 is shown in red band, which is heavier than the DNA grown in nitrogen 14, which is shown by the nitrogen 14 with the orange band, as you can see in this figure, and sediments to lower levels in the cesium chloride density gradient in an ultracentrifugation. When DNA grown in nitrogen 15, it is switched to media containing nitrogen 14. After one round of cell division, the DNA sediments half away between nitrogen 15 and nitrogen 14 levels, which indicates that now they contain nitrogen 14 50% and nitrogen 15 15%. So in this subsequent cell division, an increasing amount of DNA contains nitrogen 14, and these data supports the semi-conservative replication of DNA. Next is the semi-discontinuous replication. When the double helix of DNA unwinds, DNA replication on one of the two strands, that is 3' to 5' strand, can easily proceed continuously in the 5' to 3' direction. This behavior, where the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously, is known as semi-discontinuous replication. As you can see in this figure, the DNA replication in leading strand is continuous and the DNA replication in lagging strand is discontinuous.